Now let's move on and let's move towards an understanding of how evolution could affect the frequencies of different alleles. Remember we talked about alleles? Well, I'll just remind you what an allele is. It's an alternating form. It's different forms of the same gene located on the same place on a chromosome. So here we have two homologous chromosomes, right? So they have the same kinds of information, but they might have different alleles for each gene. So in this case, let's just say that big A codes for a green pea in the Mendelian example where you had peas and pea pods. Let's say big A is a green pea and little a is a yellow pea, right? So here we have two alleles and they're different. So the allele frequency is the total number of copies of an allele divided by the total number of alleles for the gene in the population. So in this case, we have two alleles, but we only have one dominant and one recessive. So the frequency of the dominant in this case is 50% because half of these alleles are dominant, and the frequency of the recessive is 50% because half are recessive, right? Because only one's recessive. In this case, we have two alleles. But in a diploid organism, remember, we have pairs of chromosomes, right? We have two homologs for each chromosome. So let's kind of play around with that so we can kind of understand that better. So here's your test. You have one individual. In this case, you have a manta ray. Super cool. Manta rays are super cool. Let's say you have a manta ray, and it's a diploid organism. We're looking at one Mendelian gene, single locus. How many, and by locus we mean location, right? So we have a single location in the entire genome where this gene is found. How many alleles do we have for that gene with this one organism? Well, because this is a diploid organism, it got one chromosome from its paternal side and one chromosome from its maternal side. It has one allele from the maternal, one allele from the paternal. So we have two alleles in this population of one. So what if we had a population of three? Here we have three elk walking through the meadow. How many alleles are in this population of three elk? Well, remember, each elk is diploid, so it has a chromosome, a paternal chromosome, and a maternal chromosome. How many alleles in total in this population of three? Each one has two, so the total number of alleles is 6, because each one has two, a maternal and paternal allele. I'm trying to get you to start thinking about alleles separately than the number of organisms. So here we have 4 individuals in this population, right? 4 organisms, but they're diploid. They have pairs of chromosomes, right? So how many alleles are in this population of 4? Well, just like last time, they have a chromosome from the paternal side, a chromosome from the maternal side. So for an allele, for any particular allele, if it's a single locus, they have one from the maternal, one from the paternal. So if you have 4 individuals, the total number of alleles is 8. So now let's look at this. This will be your question. You have to pause and answer this question. This is a very difficult question, huge numbers here. How many alleles in this population of 1 million flamingos? This is a massive aggregation of flamingos. It's estimated that there are 1 million in this shot. So how many alleles for this gene are in this population of 1 million flamingos? Remember, we're just asking about one particular gene. These are diploid organisms. It's a Mendelian gene. It's a single locus. How many alleles here? You can answer that below. If you guessed 2 million, you were right. 2 million alleles. So in this case, when we're talking about diploid organisms, usually the total alleles in a population is double the number of individuals in the population because each individual has a pair of each chromosome. So allele frequency, number of copies of an allele divided by the total number of alleles for the gene in a population. So when we're talking about allele frequency, we're talking about the relationship between the dominant and the recessive alleles, or whatever polymorphic alleles you have in that population. So what's a gene pool? A gene pool is a population's entire collection of genes and genetic information. And so that's kind of beyond the individual level. You have to stop thinking about a gene pool in the sense of organisms, individual organisms, but more so as a collection of their genetic information. It's the pool of all the genes without thinking about individuals, just thinking about the frequencies of each. So in this case, this individual, we'll say this is a peccary. This peccary here is got two big B's, so he's contributing two big B's to this pool of B's and little b's. This individual here is recessive, so he's contributing two little b's to the overall pool. This individual is homozygous dominant, so he's got two big B's. So if they're all contributing their own genotype, the overall total and the ratio between big B and little b is found in the gene pool, and that's separate from the individuals. So what's macroevolution? Macroevolution is evolution on a large scale, and that can mean speciation, differentiation by genuses, things like that. These are big evolutionary events. One example that we kind of started to talk about was these Darwinian finches, these finches that are found on the Galapagos Islands. Remember, those were islands that Darwin passed when he was sailing on his Beagle through all of the tropical places on Earth and gathering notes and making observations. Well, one of the observations he made was that these finches on the Galapagos Islands, they looked a lot like finches that he saw on the mainland, except they had slightly different morphologies that seemed to suit them in their particular habitat niches a little better on the island. So while these finches look like the ones in South America, the ones on the island were specialized. Maybe they had thin beaks for prying into small holes in these trees that are on the island, or maybe they had big, strong beaks that were good for breaking seeds on that island. But you could see that there were similarities. That event is called a radiation event, species radiation. In this case, there's a bunch of species that seem to be descendant from one genus or one species of mainland bird. And that's an example of macroevolution because it resulted in a bunch of different species that can't even breed with each other anymore, or they don't breed with each other anymore. So if there's macroevolution, there's got to be microevolution, right? What's microevolution? So microevolution is small generation by generation changes in a population's gene pool. So that can happen just by chance, by drift. But there's also other examples of evolution on a less than species scale. A lot of times organisms in one area will look a lot different than organisms in another area, but they're still the same species. But here's another example. So here you have Ranidomea imitator on the top left and the top right. They look very different, but they're the same species. And on the bottom, you have two different species, Ranidomea fantastica and Ranidomea variabilis. These individuals on the bottom are very toxic, and the local animals know that these colors are warning colorations. They're aposematic colorations that indicate that these individuals are dangerous, not something that you want to put in your mouth. Well, the range of this does not encompass the entire range of this species up here. So where this species up here, which these two are the same species, they just look very different. In the populations of this species that overlap with this, their coloration has begun to match these guys. And that's an advantage there because this warning code, this coding of information in color that this is dangerous, is understood by the animals in that area and predators. And so if this individual can look more like this individual, then it can communicate the information that he's potentially toxic. So don't eat me. The range of the species does not extend in the full range of the species up here. So where this population overlaps with another poisonous frog, it tends to look like that one. So in this broader population of individuals, you have this microevolution towards looking like the local colored frogs. And this is an example of mimicry. I love mimicry because it really illustrates the evolutionary possibilities in organisms. And in this case, these top two individuals, while they look very different, are actually the same species. They're just locally imitating range-restricted poisonous species of frog. And this is a great read. You can find Speciation, Frog Mimics Prefer Their Own. It's a paper, and you have all the tools now to find that kind of primary literature. So I highly recommend reading this paper, any other paper I mentioned.